Hello, everybody. Can somebody hear me? Hi, everybody. Can somebody hear me? Can you hear me? Okay. So, Oussema, you can hear me. Okay, I'm trying to call Sumir. Hi, Sumir. Are you online? Are you connected? Okay. No, I signed in. I signed in and I created the event, so you can sign out. You can sign out and sign in from your Google account, and you'll be able to connect. Now I see some people can hear me. Wissam can hear me, for instance. Okay, but yeah, I created the event. Okay, see you. Who is with us? Okay, so Oussema says he can hear and see us. Is Benjamin also able to hear and see us? Hello, no problem. I'm good. Is Ferrer is not here, right? No. Because it would be nice if we could have the speakers. And I know she has speakers. Is Mariam here?
I don't think so. I haven't seen any of it today. I don't think it was. Tracy says she's
Okay.
So she can hear you.
Okay. Okay, perfect.
No, I don't think he's open today.
So Tracy is logged in, but using probably not using.
I think she's via YouTube.
Using YouTube. Yeah, okay. So she's not able to interact. Maybe I'll switch it to the appropriate size later on. But for now, I want to. Okay, so I'm still waiting for Benjamin and Patrick to connect.
Tracy says that with the speaker, the sound is loud.
Yes, but I was talking about us here, but it's fine if it's just the two of us.
Okay, at least the sound is good.
Okay. Okay.
Now we're fixing it. Oh,
Good. I may invite, I have to invite people maybe. Benjamin, are you with us? Okay. Okay, sorry, now I see people who are muted. So, okay, Sumir is coming, so. Hi, Sumir. Hi, Oussema. I can see you.
Hi there.
I can see Oussema. Guys, it seems. It seems we have some people connected on YouTube following us, but it seems it's only after I resend an invitation to you guys during the broadcast that it's only after that I can see you and I can have the controls and mute or unmute your phone, your mic. Can you hear me, guys, now?
Yes, I can hear you, Jean-Baka. Can you hear me?
Okay.
Jean-Baka.
Yes. Yes.
Actually, Benjamin is wondering if there is a problem. He's connected. He's asking if there is a problem.
He's connected, but sorry, what?
Benjamin asking if there is a problem.
Okay, so he has a problem. That's what I hear.
No, he was asking if there's a problem.
Oh, he was asking if there's a problem.
Yeah.
I just invited him again, so he should be able to connect. He's connected, but he should be able to connect again.
Okay, Oussema, Jean-Baka, has anybody got the YouTube video channel playing because it's sending a feedback sound?
Yeah, mine is closed.
Okay, so he has a problem. No, he was asking But Tracy is following YouTube.
Yes.
Yes, I have people. I have reports from people following following the broadcast from YouTube.
Okay.
And now Benjamin. Okay,
Benjamin
Benjamin is
online?
connected now.
So
Yeah.
Benjamin should be able to present via here. Hey Benjamin. Hey.
Benjamin is here with us now.
Because it's sending a feedback sound. Okay, Oussema or Jean-Baka, is the YouTube channel playing on the background? Just pause that.
I see. I know some people are following us through the YouTube channel. So yes, it's live. I know it's live. Hello, Benjamin.
Hi.
Okay, so it seems we have, what I can see from my window is that we have 14 people watching us.
Okay.
But most of them, if not all of them, are connected, are looking at the stream via YouTube.
Okay, but they are able to see us, so it's okay, right?
Yes, they are able to see us.
Okay.
Probably, Sumir, you cannot try on the same computer to follow the stream with YouTube and with Google Plus at the same time. It would make sense that you cannot do both at the same time. But I have reports of people connected through the YouTube channel, and it's working.
Okay.
Are you going to be the first speaker, Benjamin?
Yes, is Patrick online? Because you're in two different locations.
Okay, maybe I have to invite him again. So it's Musicha.
Yes.
Does he have a Google account? Because for him to be a speaker on this platform, I think he has to have a Google account.
The email you have is for what? Is it for MLW? He has a Google account. Let me just check quickly.
Please. Because I sent him the email on his address at the Wellcome Trust, but I'm not absolutely sure it's going to work if he...
Okay. Also, all right. Okay, it's Patrick.Musicha. So the Patrick should have two P's. PP.
Musician?
Musicha at gmail.com.
At gmail.com?
But at gmail.com.
So
But it's hard. Okay, let me type it for you here so you can see.
No, it's fine. PP, double P.
Yes, yeah, just that. .Musicha at gmail.
Yeah, I sent the invitation already. So double P Patrick
Yes.
.musicha at gmail.com. Because I don't think you can connect to Google Plus from any address. It's obviously you have to connect with a Google account.
Yeah, okay.
So I sent him the invitation. Maybe we have to tell him. I'm gonna send me what's his Liverpool address? It's P Musicha.
Yeah, double P. Patrick Musicha.
Oh, no, the Liverpool.
Oh, the Liverpool.
The Malawi Wellcome Trust address. It's P Musicha at something.
Oh, okay. Can you try the same Patrick.Musicha with a single P? Because I've got two email addresses here.
Oh, the same at Gmail for the invitation, you mean?
Yes, but with a single P.
Okay. So this one, yeah. Okay. No, I was asking about his address at the Malawi Wellcome Trust, just to send him a mail saying, please connect with your.
Yeah, that one is pmusicha at mlw medical.
Musicha at, sorry, mlw.
mlw.medical, medcol.
Yeah.
.mw.
So mlw medical. medical.
OL, not A. OL.
Okay, OL, so that's medical.
Yeah, medical.mw.
Okay.
Yeah, but is it possible that for the sake of the people who have joined and waiting, for me to start as you are joining Patrick? Or you want to join him first?
Sure. No, sure. You are welcome to try. Sumir is here. We can start. You're welcome.
Okay.
So thank you for the effort you've put in this. And it's a pleasure to have you as a speaker for this first H3ABioNet series of seminars.
Thank you. Is it showing properly this the slides?
Yes, I can see the slides.
Okay.
Yes, I can see them as well, Benjamin.
Okay, so
Sorry, Sumir?
I can start?
Yes, Sumir wanted to say something.
No, Jean-Baka, please do start.
Okay, so then I'm starting then.
Yes, so I'm introducing Benjamin Kumwenda from the Malawi Liverpool Wellcome Trust Clinical Research Program. And Dr. Benjamin Kumwenda is going to talk about the transcriptomics comparisons of emerging invasive multidrug resistance in Salmonella typhimurium in Malawi. Thank you, Benjamin. You have the floor.
Okay. Thank you, Jean-Baka. I'm talking about the transcriptomics comparative project on the Salmonella typhimurium. These are my drug-resistant strains that are emerging in Malawi. And this is an ongoing work, so I don't have conclusive results yet, but I'll just share with you what has been working on so far, the type of analysis I've been trying to to do, and then where I am at the moment and the way forward. So basically, this is Malawi, as you can see, Africa, local Malawi, 15 million people with about 118 square kilometers, 1/5 of which is a lake. The Malawi Liverpool Wellcome Trust is down here in Blantyre. That's where we're based, as well as the College of Medicine. So Salmonella usually is acquired in humans through raw food or contaminated meat, and in healthy adults it causes diarrhea. But the problem occurs in children below the age of 5, and also adults who are HIV positive, where the Salmonella then this bacteria goes into the blood, causing bacteremia and the various infections, leading to complications in treating other sicknesses. So that's a critical issue mainly in the immunocompromised individuals. And this problem is aggravated by the widespread of drug resistance in the patients, which mainly is due to misuse of the antibiotics, or sometimes it's underused or overused, and usually people buy these antibiotics off the shelf, so even before they see the doctor, they've already taken them, and that also adds on to the complication. Just a little bit of some definitions. We call a pathogen drug resistant when it's resistant to more than 2 drugs. Otherwise, if it's 2 or less, we just refer to it as a drug resistant, not necessarily drug resistant. So in Malawi, there have been surveillance studies on human pathogens dating up to from 1998. And this graph here shows briefly the observations from 2008 up to 2004. But we are still collecting data, and it's being sequenced to observe the trends. So far, as you can see, Tafnurium, previously it wasn't a problem in terms of drug resistance, but has recently grown up. Its resistance has grown up, which is a problem in limiting therapeutic options that are there to treat the pathogen. This picture is taken from the work of Gordon et al., who have been involved in this surveillance, and is published in this journal here. And so in the later years, from 2006, the strains that have been collected have shown some level of emergence here. As you can see, previously all the strains were clustering together in one group, which was just drug resistant to some, I think, chlorophenicol and ampicillin, if my memory is correct. But recently, from 2002, this clade has emerged, which is drug resistant, and this also is drug resistant, and the two groups are separated by 22 SNPs, 14 of which are non-synonymous. And these two groups of bacteria were from different sources, like HIV and HIV positive and negative adults, and children, both positive and negative. But the clustering of the two groups did not, it didn't cluster based on the sources, but on the years, meaning that as we are progressing, a new clade is evolving, which points to the issue of some level of evolution taking place, and maybe eventually resulting into replacing the existing clades. And here also, this is another tree showing the clustering of these pathogens. in the Salmonella group. So it's for this reason that okay, here also I'm showing this D2, 3, and 3, 7. This cluster here is being represented by this strain D377, and in this cluster it's being represented by that. So here I'm showing the distribution of these strains over the years, and as you can see that this 377 group wasn't there before, but it's showing to be emerging in the later years from 2006. And we are yet to prove whether here indeed there's a decrease in this group, or it's just two groups separating due to evolution. So that's another question that needs to be addressed to see whether this group is diminishing or there's just a divergence occurring. So I showed you, I'll just go back, I showed you that this group, there are 22 SNPs, 14 of which are non-synonymous. And so this picture here is showing the list of those SNPs, that's 14 SNPs, some of them occurring in genes. This work was published by Oussema et al. In 2012. And so there are several SNPs scattered through in some of the genes that are being shown here, most of which are related with the energy production and the respiration, both aerobic and anaerobic. So my work basically is to investigate these SNPs if they have some biological significance in the division between these two grades. And in the previous work, I went to investigate these SNPs and look at them from the pathway point of view and from the protein structures. But now what I'm doing is to do a transcriptomics, to do gene expression and see how the differential gene expression is occurring between those two strains. So these are the questions that I'm trying to address at the moment, taking them from two different angles. So this picture here is just showing you the possible approaches that could be used to answer this question. So we have these two groups here. We have the both drug resistant. One is being represented by D23580, which has been there for a while, and this is a new one, which is being represented by D377, and I would just want to do a comparison between the two strains. So instead of comparing all the strains, we've just taken these two representative strains from both clusters. First is just to do a head-to-head genome comparison and see how similar the two genomes are. Looking at both the core and the accessory genomes. The other one was to look at those SNPs and see how the protein structure, how different they are. How did those SNPs alter the protein structures from the two strains? And then try to follow up those SNPs in metabolic pathways to see what metabolic pathways are being affected, and whether that can be related to what we're observing on the ground. And then finally, to work on the transcriptomics to look at gene expression and see if there's any differential gene expression that can be related to those different states. So here I'm showing the approach in modeling, where I identified genes which were affected by SNPs from the two strains, and then identified a template in protein data bank, and then found other orthologous sequences to do a multiple sequence alignment of the two orthologs plus the others found in public databases, just to look at how they align with the template, and then finally do the modeling, which basically copies the structure based on the alignment to come up with a resulting structure, and then compare the two structures. So that was my first approach. And out of those 14 SNPs, there were only 10 that fell in coding regions, and out of those that fell in coding regions, only 5 had templates in protein data bank, which I did for modeling. So the modeling basically were to look at the SNPs to see where it's occurring, and then follow it up in the structures, and you see whether it's altering the structures of the protein structure, and whether it's occurring on the surface or in the interior of the structures, and also finally to check whether it affects the stability of the protein structures, which is related to how stable that protein could be in an environment. So to check the stability of the structures, I use a program known as M4, which measures the minimum folding energy. So for each mutation that was introduced in the structure, I assessed the difference in the energy of the protein using that. The understanding is that the structures with less minimum folding energy are more stable and less affected by external factors. So if there is a variation in that, it can tell you what implication the SNP is bringing on the structure. So computing those energy values, it showed that in the odd cluster from which this cluster emerged, most of the values were lower, showing that possibly the structures were stable, and most of the mutations were occurring in this cluster, and where the energy values were higher. I could only speculate from this observation that possibly here the organism is still evolving to reach its stability states, and so that's why the proteins are still unstable. But apart from that, there was no any biological linkage that we could make based on this observation. So that was our first approach. Then the next one was to use metabolic pathways, in which case we use a genome and its annotation to reconstruct pathways using pathway tools, and then look at the pathways that are constructed. This figure I'm just showing, just comparing the absence and presence of, for example, where you have the red highlighted, those are enzymes that are shared, and the ones that are blue are not shared. So, but it's highly affected by the annotation that is being used. So there's need to improve the annotation so that we can reduce the number of unshared enzymes. Despite that, it was still possible to observe that most of the SNPs were occurring within specific pathways, and the tricarboric acid cycle is one of the pathways that was highly implicated, and this cycle is well known to result in reactive oxygen species. And that prior work has shown that overuse of antibiotics results in inducing the pathogen to be drug resistant. So there are about 4 genes that are related to this tricarbolic cycle. So possibly there's some degradation happening, and this is agreeing in prior work that shows that the TCA cycle when it's somehow affected, it results into high drug resistance. So that was also an interesting thing, but still the results are not conclusive. We are still trying to work and establish this with some data. And then the other part was to do a genomic comparison, looking at comparing the genomes, looking at the phages, the core genome, how similar or were they unique, and also on the plasmids. So here I'm just showing the phages that have been found, and both organisms show to have similar phages. Because phages may bring in virulent genes and maybe can contribute to the divergence of the nutrients. So this is the same list of phages that I'm showing with this diagram. They are all similar, although from this figure you can see that phages in this 377 are somehow degraded, but we suspect that maybe this may be due to the poor genome assembly that was there. So we'll try to sequence this genome and then do the comparison again and establish these differences. So in comparison, below here is 377, and on top here is the D2380. This is the other group. So we could also see that there were some gaps in the sequencing, as you can see here. This is using ACT. This is Artemis comparison 2, and I've just taken several snapshots that shows that some degradation in the sequencing. Otherwise, if we had a complete, properly sequenced genome in D377, possibly this analysis could have been more conclusive. So here is also another, as you can see here, it's clearly the genome is okay in D23, but down here there are some gaps showing that some problem in the assembly. However, so it seems like this chunk here may be missing in this. So we need to establish whether this is a real difference or it's a problem due to the sequencing. And that's another figure as well. So what I'm busy doing now is the last part of the question, where we are looking at the gene expression, which has been studied for many years, and in Salmonella it has been well described. The idea is to understand how these genes get expressed differently as Salmonella is ingested and moves through the mouth, through the stomach and the small intestines, encountering different conditions in all those situations. And also as it gets inside the host cells, and as it encounters different stresses, such as encountering the bile in the stomach, encountering the aerobic conditions in the small intestines, and all those factors, they induce different types of genes. So this figure here is just showing the different environmental conditions that Salmonella goes through into stomach, small intestines, and then the different pressures that it goes through. This is within the mammalian cell, in the extracellular life cycle, and this is the intercellular life cycle. What happens when now Salmonella gets into the cell, what other conditions are experienced, like low magnesium, the change in acidity, all those factors. Here is just showing the different genes that are been known to be expressed in those both different conditions, extracellular and intercellular, and then some which are expressed in both. So we are following up this approach by Herbert et al. 2011 to do the gene expression mimicking all these conditions in a mammalian organism to investigate what genes are expressed, and depending on how those expressions differ, then we'll look for whether there are SNP differences or whatever, whether it's regulatory differences, and then follow up on those. If there are SNP differences, then we move on to do some modeling and see whether there are changes in the structure or what metabolic pathways are involved, so that we can link the observation to the biological situation that we are seeing in Malawi. So in summary, we have investigated the SNPs separating the two Salmonella drug-resistant clusters using pathway tools, metabolic pathway analysis, and also using the minimum folding energy. And then also now I'm busy working on the growing these salmonellas and then isolating the RNA, and then we're going to sequence the RNA so that we can see the differential gene expression. So that's the end of my talk. These are the people that I want to thank: Jay Hinton at the University of Liverpool, and Rob, Malawi Wellcome Trust, Dean Everett, Kirsten Kroger is the one I'm working closely with on the transcriptomics, and Chisomo. So, and thanks to H3ABioNet and Malawi and University of Malawi for supporting me. And thank you everyone for listening.
Thank you, Benjamin.
Yeah.
Thank you, Benjamin.
Interesting talk. We may now proceed with questions. So I unmute everybody. Those who are following us on YouTube, you cannot ask questions if you're just following the video. So to ask questions, one has to log into Google+. Oussema, you wanted to say something?
Yes, I want just to thank Benjamin for his interesting talk, and I want just to centralize the questions through Google+. That's just what you want. Just said. It's okay. So we have to centralize them through Google+.
Okay. Yeah, people connected to Google Plus can also. Oh, sorry. So I see some discussions on Google Plus. Unfortunately, Tracy, we could not make the slides full screen because it's screen sharing. It's a limitation of the screen sharing apparently with Google Plus and Google Hangouts. We tried during our test on Wednesday, but it wasn't possible to make things full screen. Okay, so we had around 11 to 14 attendees. Has anybody got a question? Otherwise, I have one.
Is it not possible for them to text the question to you and then you?
Sure.
Forward
They can,
and then we.
even if they are following, even if people are following through YouTube, they can on Google Plus they can text questions. I wanted to ask you, Benjamin, if you had investigated other genomic differences than just SNPs, like translocations or multiple repeats, tandem repeats, etc.
Unfortunately, Salmonella is highly conserved, so we don't expect much of those. But also, the sequence that I was showing has got some issues in terms of its assembly. So we are thinking that whatever we would get, we may not be confident of the result. So right now we're just sequencing it using PacBio and hoping to get a better assembly, which then we can use to do those comparisons. So maybe it's something that can easily be done when we have another. For the D23, the sequence is fine. The genome sequence is fine. The quality is good. But for the 377 cluster, the sequence is not very good. So we are sequencing it now.
Mm-hmm. Okay. I have a second question. It's quite interesting. It's quite nice to see that you detected that in those emerging clusters, minimum folding energy was higher, and you indicated that maybe it's because these emerging clusters are still under quite a big evolutionary pressure, and they are finding their best fits or the best proteins. It's quite interesting. And have you tried to resequence in the same emerging clusters after 2 years, or to see whether we have some tangible evolution?
Yes. So some of the in Patrick's project, he also he sent samples to Sanga for sequencing, but they are not ready up to now. So we hope that when those sequences are ready and assembled, then we can use them to investigate. I mean, to establish what we are observing further. Because so far, although the data is showing something, it's still limited to be very confident on what we are observing.
Okay, thanks. Other questions? On this work, web link itself. Okay, apparently people want me to share the web link again, but I don't understand if they can talk to us. They can, Vicky, if you can talk to us through Google Plus, you should be able to just click on joining the event called H3ABioNet Webinars Launch. I'm going to try send the link again. But, okay, Patrick, I think you can get ready. Patrick, can you?
Hello.
Can you hear me? Yes. Hello, Patrick. I'm going to toggle the video. Thank you, Benjamin. So I'm going to give the video now to Patrick.
Okay, thank you.
Patrick, can you please try to share, to click on sharing your screen?
Yep.
With us. On the left, you have screen sharing. Yes, hello, Patrick. I'm going to toggle the video. Thank you. So I'm going to Patrick.
Okay, thank you.
Right. Are you able to see the presentation?
Yes, but can you close the left side?
Okay.
Patrick, I cannot see your Okay. It's fine. Patrick, can you try maybe to go full screen or to go to presentation mode on your, to enter presentation mode?
Like this? Is that fine?
Try by typing. Try by typing.
Maybe on the.
Yeah.
Presenter view.
Presenter view.
Okay.
Is the,
But we tried.
Hello?
But we tried on Wednesday unsuccessfully.
yeah.
This
Yes,
was
Patrick, are you full screen now?
Yes.
You are full screen, okay, but not for us. So it's the same as during our test. Are you full screen on your screen?
Yes.
Patrick, okay, so yeah, it seems we cannot.
Just try to increase the window. Just drag it down to the full window.
And if you go on the left, lower left corner of your screen, if you click on the presentation view slideshow, yeah, I think this freezes for us. It won't.
I'm on slideshow. Can
Yeah.
you see it? No.
No, no, no. Unfortunately, we tested that already, and it wasn't working. So please revert to the window you had, not slideshow, but kind of editing window. And yes, we can follow from this. But try to make sure that all your slides fit in the window.
Right.
Maybe the bottom right now, the bottom is not viewable. If you can decrease the size. Yeah, maybe drop this down. Yeah, maybe like something like that. Okay.
Okay.
Okay, you have the floor. Vicky has a question on the chat, but unfortunately, I think we have to proceed with Patrick because it's already 1 p.m. and maybe Benjamin can answer.
The second talk, we can go again and listen to Vicky's question.
Okay, okay. After Patrick's presentation, you mean?
Yeah, I think. So, Patrick, go ahead.
Right, thank you.
Thank you.
Fine. So, the title of my presentation is on diversity of mobile genetic elements and antimicrobial resistance genes in 3 Salmonella cells. Before I go into the actual presentation that I have here, I'll briefly talk about my project, the whole project. So my PhD project is looking at trends and the genetic determinants of antimicrobial resistance in enteric bacteria. And the 3 enteric bacteria that we are looking at are Salmonella, E. coli, and Klebsiella. Basically, what we are trying to do is we will use 2 approaches. One is going to be a mathematical modeling approach where we are going to model the trends of antibiotic resistance based on the phenotypic data that has surveillance data that has been collected at the Malawi Liverpool Wellcome Trust for the past 20 years. And then we also analyzing genome sequences of isolates, some of which for E. coli and Klebsiella we have sent for sequencing. And for Salmonella, we have isolates that were already sequenced, and those are the ones that I've been working on. So the presentation here is on some work that I have done on Salmonella, and whose isolates were already sequenced. Fine. So Benjamin has given a comprehensive background of Salmonella and the infections with which they are involved. But in addition to that, I have some background here. So Salmonella belongs to the family of bacteria called Enterobacteriaceae, commonly known as enterics or enteric bacteria. It has two species, Salmonella enterica and Salmonella bongori, and the species are divided into serotypes. Salmonella enterica, which is the one that is responsible for the majority of the human infections, is divided into 6 subspecies and over 2,500 serotypes. But the disease causing Salmonella are categorized into 2 broad groups known as non-typhoidal Salmonella and typhoidal Salmonella. And in sub-Saharan Africa, the major serovas or serotypes that are responsible for Salmonella disease are non-typhoidal Salmonella disease are Salmonella typhimurium and Salmonella enteritis, whereas typhoidal Salmonella disease is largely caused by Salmonella typhi, and in other countries by Salmonella paratyphi. So in recent years, the management of Salmonella infections has been complicated due to the emergence of drug resistance. And as Benjamin said, in our case, we define drug resistance as being resistant to at least 3 antibiotics. And the common antibiotics for treating Salmonella infections are chloramphenicol, ampicillin, and cotrimoxazole. So if we see resistance to those 3 antibiotics, then we classify that organism as being drug resistant. And classification of the Salmonella typhimurium, drug-resistant Salmonella typhimurium, that is responsible for the invasive disease in sub-Saharan Africa, shows that the majority belong to a unique Madloga sequence type, which is designated as ST313. But the antibiotic resistance is encoded on a composite element, which is known as TN21, and this element is inserted on the Salmonella typhimurium virulence plasmid. Now, Benjamin talked about the Salmonella clustering into 2 lineages by looking at the genomic sequences, sequence comparisons. But then when you look at it from the plasmid's side, we've also noted that these 2 clusters, the same clusters that we see for the genome sequences, also appear when you compare the plasmids. Those 2 clusters, both of them have a virulence plasmid, and this virulence plasmid is the same within each cluster, but different between the clusters. The difference exists in the TN21 element, so they differ on the position, but also on the resistance genes that they carry. So one cluster, which is now the dominant cluster, carries the chloramphenicol resistant gene, which is known as CAT gene, whereas the other one does not carry that chloramphenicol resistance gene. But also we have seen that the plasmid, the TN21 element region in the cluster which carries the chloramphenicol resistant gene is unstable, suggesting that it can there is a high likelihood of further microevolution of the plasmid, unlike in the other cluster. Now, the other Salmonella typhi, the other serova, which is Salmonella typhi, this is the dominant cause of typhoid fever, and typhoid hasn't been a big problem in Africa, unlike as compared to non-typhoid or Salmonella disease. Typhoid has caused much of the problems in Southeast Asia. However, recent surveillance data show the emergence of Salmonella typhi epidemics across sub-Saharan Africa. And further to that, the isolates that are responsible for this epidemic are also but drug resistant, and they are resistant to the commonly antibiotics that are being used in the hospitals. And beyond being resistant to those antibiotics, which are driven by the acquisition of resistant the plasmids, which have been talked about, there are also some resistance. There is some resistance that is being driven by mutations on the chromosome, and this is this resistance is to the second-line antibiotics, which include fluoroquinolones, and that is causing a big problem because already there is resistance to chloramphenicol, ampicillin, and cotrimoxazole. And if resistance spreads or to fluoroquinolones, then that will limit the treatment options for typhi disease in Africa. In Malawi, we have seen 3 epidemics of Salmonella. The first one was between 1999 to 2003, and this epidemic was caused by Salmonella enteritis. And after that, there was an emergence of a Salmonella typhimurium epidemic. This from 2002, and this has persisted up to now. Salmonella typhimurium is still a big problem in Malawi, except that over the recent years, after the introduction of free antiretroviral therapy to HIV positive people, there's been a decline in the incidences of Salmonella typhimurium, and from 2011, now we have seen an epidemic in Salmonella typhi. One thing that is common about these epidemics is that they are all preceded and characterized by images of drug resistance, but also the phenotypic tests on antibiotic susceptibility shows that all the 3 epidemics have similar antibiotic resistance profiles. Now, the question that I had for this week was that since we are seeing some commonalities in terms of every time we see an epidemic, it is preceded and characterized by images of drug resistance, but also the resistance profiles are the same. The question that I was asking was, the mobile genetic elements and the antimicrobial resistance genes in these 3 serovas the same? So I collected isolates that were previously sequenced at Wellcome Trust Sangha Institute. These were typhi and typhimurium isolate reads, and they were sequenced using the Illumina MySeq platform. And on top of that, I also managed to get one typhi strain, which is 10.36.491. This one was resequenced using PacBio platform. And then I created pseudo-reference sequences because I was interested in the mobile genetic elements, and therefore I could not map them to a chromosome reference sequence. So I had to create a pseudo-reference sequence that comprised of the chromosome as well as the plasmid sequences. So I had two drug reference strains. One was CT18 for Salmonella typhi, and another one was D23580 for Salmonella typhimurium. CT18 was an isolate that was collected from Vietnam in 2000, and it was sequenced in 2001, and it is a drug-resistant strain. So it is resistant to chloramphenicol, ampicillin, and cotrimoxazole, among other antibiotics. And this strain was found to have two plasmids. One of the plasmids, which is called PHCM1, is responsible for the drug resistance. All the resistant genes were encoded on that plasmid, whereas the other plasmid, which is known as PHCM2, was cryptic, so its function was not clearly known, but it was never associated with antimicrobial resistance. Salmonella typhimurium strain D23580 is also a drug-resistant strain of Salmonella typhimurium. This was isolated in Blantyre, Malawi, and it was sequenced in 2004. Again, for this one, resistance was on a virulence plasmid known as PSLT-BT, but it has 4 plasmids. The other 3 plasmids are also known to be cryptic, so their function is not clearly known, but it is not associated with the antimicrobial resistance. The resistance genes encoded on PHCM1 and the PSLT-BT are similar, and you could. I was thinking that probably because these isolates co-circulate in the host, there could be a possibility of horizontal gene transfer, considering that the genes are on a plasmid which can easily move from one bacterial cell to the other. And then, so I mapped my reads to these pseudo-reference sequences, and then analyzed the alignments and did phylogenetic analysis to determine the genetic relatedness of the mobile genetic elements as well as the genes. And because of the results that I found, which I'm going to show you in a few moments from now, I had to go further to do a genomic comparison of CT18 chromosome sequence and the PacBio sequence strain of 10.36.491. So when I did the mapping, I found out that the genomes, the reads mapped to the genomes, but only a few reads mapped to the plasmid legions. And so this is for typhi. The picture that you are seeing is for typhi. The top panel represents the the plasmid for typhi reference sequence. This plasmid is PHCM1, which is the drug-resistant plasmid. And the bottom panel represents the sequence from one of my isolates. So the reads did not actually map to the plasmid, only except for 8 blocks. So I found 8 blocks to which the reads mapped. And these 8 blocks formed about 24 kb. Yet the plasmid PHCM1 size is 218 kb. So that was about 11% of the plasmid sequence. And to me, what that meant was that these isolates do not have that plasmid. And then looking at how the reads mapped to the second plasmid, no reads were mapped to the second plasmid, so that one was completely empty. However, when I did pairwise comparison of one of the isolate mappings and the PHCM1, as I am showing here, and also did some function analysis by blasting the block sequences against the NCBI database, I found that these blocks contained the resistant genes and also some mobile genetic elements, particularly of the type IS1. And the message was that the plasmid is not there, but the isolates have the genes. So it meant 2 things to me. Either the genes were on the plasmids, but they were on the chromosome, but they had to map the plasmids of mapping. Mapping, you map to reads map to where there is some similarity, and their similarity from the isolates was found on the plasmid. So they mapped it to the plasmid, when in actual fact they might not be on the plasmid but on the chromosome. Or these isolates contain a totally different plasmid that we don't know. So I tried to do sequence comparisons with the other published Salmonella typhi plasmids, but I found that all of those were absent. And then also considering that these are co-circulating with the Salmonella typhimurium, then I did a comparison with the PSLTBT, the plasmid for the virulence plasmid carrying the resistance genes for Salmonella typhimurium. I also found that this was absent. So it's when we obtained a PAGPI sequence of one of the typhi isolates, and sorry, so there is some image here that is not, that is removing, fine. So this data and the image that has just gone out of the slide are actually trying to ascertain that the blocks that I found were actually associated with the antimicrobial resistance. And we noted that the isolates that did not have the blocks were actually most of them sensitive. So I had the 30 isolates that in which I did not see these blocks, and out of those 30 isolates, 30 isolates that were sensitive, out of those 30 isolates, 28 did not have the blocks, and out of 67 isolates that were deemed to be resistant to all the 3 antibiotics, 63 of them had all the blocks that I had found. 3 had 7 of the blocks and only 1 had 1 of the blocks. So looking at the data, it was clear that these blocks were in some way associated with the antibiotic resistance in the isolates. So when I did a pairwise comparison, now this time around I only compared the chromosome sequence of CT18 and the chromosome sequence of the PAGPI sequenced strain 1036.491. And what we found was that there was an insertion on the chromosome of 1036.491. And when I extracted this insertion and did a mapping of the reads to this insertion, we found that the blocks actually were part of this insertion. So what it meant was that in the Malawian isolates, resistance on the typhi, Salmonella typhi isolates is not on a plasmid, and the isolates do not have a plasmid, but rather it's on a chromosome, unlike in Salmonella typhimurium. So this table here, I'm just trying to show a comparison of what I've found in Salmonella typhi as well as in Salmonella typhimurium. So first of all, we know that the Salmonella typhimurium has a virulence plasmid PSRT-BT, which is carrying the transposable element TN21 and the resistance genes. Whereas in S. typhi, there is no plasmid that we have found, but there is a composite transposable element, IS1, and this one is inserted on a chromosome. But then it carries similar resistance genes to those that are carried on the Salmonella typhimurium plasmid. So in summary, we are saying that the plasmid, the isolates, the Salmonella typhi isolates have no plasmid, but the resistance genes are encoded on a transposable element. The genes are similar. Now, from the work that I was doing, from a bioinformatics point of view, there is probably always a question of whether, if you want to do reference mapping, reference-based mapping, or you want to do de novo assembly, which one should you go for? And there was one disadvantage that I noted in doing reference-based mapping, and in that features can be located on a, it can give you a wrong locus of a feature. So for example, if the plasmid was present, but the transposon was, the mobile genetic element was still located on a chromosome, because in the reference sequence it is on a plasmid, then it would have shown that I have a plasmid which has the mobile genetic elements, when in fact the mobile genetic elements are not on a plasmid, but rather on a chromosome. So this is one of the things that I saw in terms of bioinformatics methods. And therefore, in conclusion, what we are saying is that Salmonella typhi and Salmonella typhimurium in Malawi have similar resistance genes, but they have these resistance genes are carried on different mobile genetic elements, and moreover, the mobile genetic elements of Salmonella typhi are located on a chromosome and not on a plasmid as it is with the Salmonella typhimurium. And therefore, resistance in that we are seeing in typhi is not as a result of horizontal of exchange of genetic material between Salmonella typhimurium and Salmonella typhi, but rather it's a result of independent evolution and expansion of the drug resistant clones for each of the solvers that we are looking. And so this is the conclusion that I get from the work that I have done so far. This work is continuing because in the title I'm talking about 3 servers. So we have done a comparison with Salmonella typhi and Typhimurium, and then we are looking further at Salmonella enteritis to see if the mobile genetic elements in there are similar with either the ones in Salmonella Typhimurium or Salmonella typhi, and if the genes are similar or different, and whether the resistance is due to horizontal gene transfer or each of the resistance in each of the servers is independent. So thank you. That's the end of the presentation. I would like to thank my supervisors, H3ABioNet, who are the principal sponsors of my PhD, and also the Southern Africa Consortium for Research Excellence, which are supporting my modeling work. Thank you.
Okay, thank you, Patrick, for this very interesting presentation. Are there any questions? Sorry, Oussema, I muted you. Yeah. So questions can be asked also via Google+.
Yeah.
Maybe my question is about your conclusion. I'm not an expert on Salmonella and those small organisms, but what makes you think that there is convergent evolution and not that there has been a transfer of genetic material between these 2 species?
Right. So, if it were a result of genetic transfer of the genes, the resistance genes, then they would have been, despite that the genes are similar, but they would have been carried on a similar mobile genetic element. So the fact that the genes are the same, so the resistance that we are seeing is very similar, but that they are carried on a different mobile genetic elements, then it means that the mode of acquisition of the genes was different in the 2, because they could have, the same mobile genetic element could have moved from one serotype to another serotype, but we are seeing different mobile genetic elements. So there is no there's nothing to show, or there's no carrier of the mobile genetic elements from one serotype to the other serotype that we are seeing.
Okay, thanks. Do we have other questions?
Maybe I have one but naive question. Among all your mobile genetic elements, you investigated only transposons, or you extended these to the others like phages and so on?
So we only looked at the transposons because these are the ones that have been documented to be carrying the resistance genes. But also, the primary target were the resistance genes, and then we are looking at where are the resistance genes. So as I said, that the pseudo-reference sequence that I created, I did not just look at the plasmid. I looked at the pseudo-reference sequence, which was created by combining the chromosome sequence as well as the plasmid sequences. And that means that if there are phages that we are carrying resistance genes, then we should have seen them.
Yeah.
Yeah. So in this case, the analysis excluded phages from being responsible for antibiotic resistance.
Okay. So are there some other questions?
Sorry?
No, I'm just asking if there are other questions.
Oh, okay, fine.
There was a question to Benjamin. Benjamin is still with us. I'm unmuting you, Benjamin. People asked you whether you had explored whether there is a high genetic pressure around the areas of the genome where you have those 5 proteins that you looked at.
No, I did not do that, but I think it's a good point. I will try to include that in my analysis.
Okay. Vicky, do you still want to ask a question? Yes.
You have those 5 proteins that you looked at.
No, I did not do that, but I think it's a good point. I will try to include that in my analysis.
I'm sorry, maybe it comes from Vicky's session or from...
Vicky, do you still want to ask a question?
Yeah, what we could hear was coming from Vicky's
hi.
session. Okay.
No, I did not do that, but I think it's a good point. I will try to include that in my analysis.
I'm sorry, maybe it comes from Vicky's session or from...
Vicky?
Yes.
Sorry, Vicky.
I think the question has been asked already.
Yeah, what we could hear was coming from Vicky's
hi.
session. Okay, it's weird. We have some lag from... Okay, it's weird. Sorry, Vicky, I had to mute you because it seems, I don't know, we have an echo from your side. But yes, Vicky's question was about whether Benjamin had explored if the region where those 5 proteins are is already under selection pressure. So Benjamin already answered. Well, thank you, Patrick, and thank you, Benjamin, for this this first seminar. Yes, another question? Just maybe from Vicky. Yes, Vicky, we can hear you. Vicky, we can hear you now.
Yes, I think the question has been asked because I was trying to.
Now we can no longer hear you. We hear the beginning of the question. You said the question has already been asked. Yes, Benjamin answered that he has not done so, and your point is a good one. Okay.
Okay, so do we have other questions, or it's time to conclude?
So thank you everybody for attending. It's true, Sumir noticed that the YouTube and Google Hangouts are playing simultaneously with half a minute lag, so it's a big lag. So that's why we had this problem of echo sometimes. But thank you for being the guinea pigs in this experiment, and other seminars will follow soon, probably every second week.
Okay. Thank you.
Yeah. Thank you all. On behalf of the H3ABioNet Research Group, I would like to thank our two volunteer speakers, Patrick and Benjamin, for their interesting talks, and would like to thank you all for your attendance in this inaugural series that will be, as Jean-Baka said, renewed, we hope, every 2 weeks. So thank you all.
Thank you.
And Jean-Baka for managing this.
Yeah, thank you. No problem. Goodbye, everybody.
Goodbye.